All right, on the last slide we were looking at HIV, and HIV is included in this list. But the first thing that I'm asking you to do here is identify those viral diseases that we have a vaccine for. So ask yourself, is there a vaccine for the common cold? Have you ever heard of that? Is there a cure for the common cold? No. Influenza? Yeah, there's a vaccine, but you have to get a different one every year. You can't vaccinate against the flu and have it last your whole life. HIV, AIDS? No. For all of these things, they're trying to create vaccines, but they're not successful, and definitely not successful at creating vaccines that are 100% effective. Even the flu vaccine is not 100% effective. Chickenpox? Yeah, there is a vaccine for chickenpox. Hepatitis B? Yeah, there is a vaccine. West Nile virus, Zika virus, Ebola virus? Nope. So if you take a look at which ones we have vaccines for and which ones we don't, hopefully you can see some commonality between those, the ones that are in each one of those groups. The ones that we have vaccines for are primarily these DNA viruses. And the reason for that is that DNA does not mutate as quickly as RNA viruses do. So it's these RNA viruses that are the big problem. RNA is less stable than DNA, therefore it mutates at a higher rate, and therefore it evolves at a higher rate. And so because it changes so quickly, the vaccine doesn't work anymore, or your immunity to the virus doesn't work anymore because it changes and your immune system can't recognize it. Because remember, that's how vaccines work. You get vaccinated, the vaccine teaches your immune system to recognize the virus, but then if the virus changes, like these RNA viruses do, they change very quickly from year to year, your immune system can't recognize them anymore. So that's a big problem. There are viruses too that are infecting our crops and domestic livestock. So, it's just not human diseases that are a problem with viruses. Just like the very first virus ever discovered, tobacco mosaic virus, it's a plant virus. So there are viruses that infect our food supply. So that's the other problem for humans. And I just put the symptoms of Ebola here because arguably Ebola, along with HIV, are probably the two worst viruses here in terms of the symptoms and how nasty they are. And we're lucky that they're not communicable from just casual contact. If they were, then they would spread like wildfire and we would all be in big trouble. Viruses aren't the only infectious agents that that get their nucleic acids or genetic material into cells and disrupt cell function in that way. We also have these things called viroids. But viroids don't infect people, they don't infect animals, they only infect plants. And what they are what you could refer to as just little pieces of naked RNA. In this image, we're seeing viroids. This is a scanning electron micrograph of viroids, very, very, very highly magnified. And you can see that some of them are in the form of a ring, and some of them are linear. But all they are is a short little piece of RNA. There is no capsid surrounding them. There's no protein associated with that RNA. It's just naked RNA. And it gets into cells when the tissue or the cells are damaged. So they have no way of getting into healthy cells. But if the plant is damaged, like say for example somebody's weed eating around the bottom of a tree and hits the tree and takes the bark off the tree and damages the tissue, then a viroid could get inside that damaged tissue. Just like viruses, viroids replicate inside cells, and in the process of replication they interfere with the host cell function. So they hijack cells just like viruses hijack cells, but they have no capsid, and they infect only plants. So in this experiment, this is showing the procedure that was followed to discover that viroids exist. So the compounds that end in ASE, as you should remember, are enzymes. So protease, lipase, RNase, and like any good enzyme name, the name of the enzyme tells you what it does or gives you some indication of what it does. And in this case, these are hydrolytic enzymes. If you'll recall, hydrolysis is how polymers are split back into monomers. So in other words, hydrolytic enzymes break things down. So in this case, proteases, the protease breaks down proteins, lipase breaks down lipids, RNase breaks down RNA. What's in the test tubes here is sap from an infected plant. So if you take sap from an infected plant and put it onto an uninfected plant, it will infect that plant. So over here with the no treatment, I'm just using that as an example, so that the disease can be passed on from an infected plant to an uninfected plant by the transfer of sap. And of course there would have to be some tissue damage too. They didn't show that in the diagram, but they probably made a slight cut in the leaf before they transferred the sap. Possibly. The purpose of no treatment is as a control group, right? So we have 4 experimental groups here, and you have to also assume from this experiment, or assume in this experiment, that they're repeating this numerous times. So these really are groups. There isn't just one plant that's being infected here. There are multiple plants in each group. But this is the control group because there is no treatment. It gives you something to compare the other groups to. If it wasn't for the control group, you would have nothing to compare the other groups to. That's the purpose of the control. So each enzyme in this experiment hydrolyzes a specific class of compounds, like I pointed out, and the blotches on the leaves represent transmission of the disease. In other words, the only plant that did not contract the disease in this case is this one, and it's the one that was treated with RNase to destroy RNA. So that showed that it's RNA only that infects plants, that passes on these viroid-caused diseases. If it was caused by a bacteria or a virus, then both protease and lipase would have stopped transmission. But they didn't. So the bottom line is that viroids are naked RNA, just little molecules of RNA that infect only plants and can only get into plants if there's tissue damage. So here we're looking at prions, and prions, kind of like viroids, only infect animals. Viroids only infect plants, and prions only infect animals. And just like just like viroids are genetic material only, RNA only, prions are protein only. And they infect only animals. And it's transmitted by eating the tissue of an infected animal. So in other words, if there's a cow that's infected and you eat a steak from that cow, then you could be infected by the prion. But there's no capsid to help them gain entry, so that's why you have to eat the prion in order to get it into your system. What it does is it gradually deteriorates your nervous system. And it does that by interfering, or by building up. These things called amyloids build up in your nervous tissue and interfere with primarily brain function. So that's why mad cow disease, it drives the cows mad. And we can catch mad cow disease. If we eat the prion that causes mad cow disease, we can get mad cow disease. What causes prions, or what allows prions to do the damage they do, is what they are misfolded proteins. So they are a protein that's supposed to be in a cow or supposed to be in a person, but they fold the wrong way. If you'll recall, proteins, when they're made, when the amino acids are strung together to make a protein, they fold in a particular conformation, a particular shape that gives them their function. And when it comes to prions, they're misfolded. So in the diagram here, the green represents normal and the red represent misfolded. And once they misfold, and this is the nasty part of them, it kind of causes a chain reaction. They cause other normally folded proteins to also misfold. And these are all the same kind of proteins. But as soon as one misfolds, then it causes others to misfold. And then on top of that, once they misfold, they stick together and form these fibrils that are known as amyloids. And so if you look at the brain of an infected cow or an infected person, you can see the buildup, these splotches in the brain tissue of the buildup of amyloid. They call them amyloid plaques. And then the amyloids, these fibrils, will break apart. That's fractionation. Fractionation just means break apart. And the pieces will, each piece that breaks off of the amyloid will go on to recruit other normally folded proteins to become misfolded. And so it's, like I said, it's a chain reaction, or a positive feedback cycle too, that causes the buildup of amyloids in the brain and the damage of brain tissue. And that's what causes the symptoms. So the disease we're looking at here, like I said, is mad cow disease, but the technical term for mad cow disease is bovine, which refers to cows. Bovine spongiform encephalopathy, BSE. You can see why you'd rather use the acronym. Another disease that's a human disease is known as Kuru. And that infects people, tribes in Africa, that typically part of their death ritual is to eat the brains of the dead person. So again, you have to eat prions in order to get them into your system. And in the practice of eating the brains of their dead comrades, Kurus passed from person to person in those tribes that practice that death ritual. The only absolute diagnosis is to look at the brains of people that have been infected, but after they're dead. Post-mortem means after they're dead. So there's no way to diagnose these diseases before a person is dead. They can only discover that they were infected with mad cow disease, for example, after they die. So in the figure here, it's the left brain that is not normal. It's all, you can see that it's kind of shriveled and darker, and that's because of the buildup of amyloid in that brain tissue. So just to summarize and make sure you know the difference between viroids and prions. Viroids are naked RNA, little pieces of RNA that infect only plants, and prions are proteins, misfolded proteins, that build up in the nervous tissue of only animals.